Good morning, it's August 12th, and this is your daily brief in science. Here's everything you need to know. A programmable DNA origami nanosyringe is shown to do more than carry cargo. It acts as a trigger to start DNA hybridization chains, RNA transcription, and DNA-catalyzed reactions at precise times and places. The device achieves controlled transport while preserving membrane integrity, enabling temporal and spatial regulation of biochemical processes inside synthetic cells. Through spatiotemporal control, the nanosyringe delivers cargo at defined moments and specific sites within synthetic cell systems. Membrane perforation is driven by downward needle sliding, with pore formation confirmed by single-channel recordings and conductance kinetics that match natural pores. DNA origami systems enable membrane-localized hybridization chain reactions within giant unilamellar vesicles, producing a ring-shaped DNA cortex near the inner membrane after needle penetration. The DOS is compared to microneedles used in assisted reproduction, highlighting its membrane-centric operation, potential for automation, modular cargo tethering, and prospects for expanding tethering chemistries. A hexagonal lattice variant that occludes central channels demonstrates that central channels are the main transport route, with minimal disruption to the periphery. The work builds on prior contractile injection system research and related nanotechnologies, drawing on foundational chemical biology and nanotech studies. The DOS consists of two 14-helix DNA origami bundles connected by a gold nanoparticle, featuring a base and a needle with central transport channels and 7 cargo conjugation sites at the tip. The system is reversible. Retracting the needle restores membrane integrity, and fluorescence recovery after photobleaching and current measurements confirm resealing post-actuation. Activation relies on DNA strand displacement to drive forward needle penetration and backward retraction, with a membrane anchoring base and a movable needle connected by a reversible sliding mechanism. Transport occurs through the needle's central channels for small molecules and via tethered cargo at the tip for larger biomolecules, showing size and charge selective transport in experiments with SRB, CF, and dextrin. Introgression and hybrid genomes are shaping adaptation across plants, fish, primates, and humans, underscoring its broad evolutionary role. Meta-analyses indicate introgression is more common than once thought, which highlights the need for standardized reporting across studies. Robust methods for local ancestry inference, detection of introgressed loci, and simulation-based assessments are central, including ABA-BABA tests, hidden Markov models, pseudo-likelihood approaches, and multi-species coalescent methods. Foundational reviews and books from the late 1990s to mid-2000s laid the core concepts and frameworks for studying natural hybridization and evolution. The collection presents hybridization as widespread and impactful, challenging the idea that it is rare in animals. Genomic and statistical tools such as D-statistics, QPADM, F-statistics, admixture graphs, and phylogenetic networks are essential for detecting and characterizing introgression. Recent work emphasizes repeatable evolutionary patterns in hybrids and reinforces calls for standardized reporting to enable accurate cross-study comparisons. Human evolution remains a focal point, with Neanderthal and Denisovan introgression illustrating broader implications for speciation and extinction dynamics across taxa. Empirical findings show ancient and ongoing hybridization driving adaptive radiations, as in cichlids, evidence of selection on admixed genomes, and the influence of recombination on introgression outcomes. A new study in quantum shows that entanglement scaling in systems of interacting fermions depends on the Rényi index, meaning traditional Rényi-based diagnostics can miss high entanglement in certain states. Researchers demonstrate that the second Rényi entropy does not always mirror von Neumann entropy, as rare particle number sectors can drive the second Rényi up logarithmically, even when von Neumann entropy grows much faster. This reveals limits to using the second Rényi as a universal proxy for entanglement and highlights the need for more nuanced tools in many-body quantum systems. The work builds on symmetry-resolved entanglement research and generalizes findings to more complex quantum states, suggesting the transition is potentially more widespread than previously thought. It points to rare, fixed particle configurations that dominate Rényi calculations and mask true multipartite entanglement, prompting new analytical approaches for free fermion and related systems. The authors propose a symmetry-aware lower bound on von Neumann entropy based on charge-resolved Rényi entropies, offering a practical protocol to diagnose anomalous entanglement scaling from experimental data. The Tennessee study by Barghouti and Del Maestro shows that entanglement scaling can shift from power law to logarithmic growth as the Rényi index changes, extending beyond simple spin models to interacting fermions. Overall, the findings challenge the universality of the second Rényi entropy as a proxy for von Neumann entropy and provide a practical method to gauge entanglement scaling from accessible measurements. The work underscores how symmetry-aware analysis can reveal divergences between von Neumann and Rényi entropy within the same system, enabling detection of underestimation without full-state tomography. Researchers report rapid entanglement across 2 to 6 partite states and extend the approach to 10-qubit entanglement, indicating scalability and robustness to certain errors. The work suggests PT-symmetric devices could serve as versatile platforms for creating and manipulating diverse quantum resources with potential impact on quantum information technologies and hardware development. The acceleration in entanglement generation is linked to the distinctive properties of PT-symmetric systems near exceptional points where spectral coalescence occurs. This study reflects cross-border collaboration involving Chinese institutions and international partners in quantum information science. The findings contrast with passive PT-symmetric approaches and show successful entanglement preparation even when gain and loss are not perfectly balanced. PT-symmetric systems enable faster entanglement preparation with higher fidelity, addressing the traditional speed-quality trade-off. The method remains resilient against non-resonant and coupling strength errors, broadening practical applicability in quantum devices. The research identifies and addresses a trade-off between entanglement degree and fidelity, effectively overcoming it. Researchers include Bingbing Liu and Shilei Su from Zhengzhou University, Henan Academy of Sciences, RIKEN in Japan, and Peking University, signaling international collaboration. Awicli is the first once-weekly basal insulin approved for adults with type 2 diabetes and is now available in the United States, delivered via the Flex Touch pen with guidance on weekly dosing, device use, site rotation, and safety. Novo Nordisk operates a patient education center at novacare.com, offering resources, affordability support, and tools to help eligible patients reduce out-of-pocket costs. Prescribing information and safety data are accessible through awicli.com and NovoCare resources, with a detailed prescribing document linked in the release. The regulatory history includes a prior FDA complete response in mid-2024 due to hypoglycemia concerns, with an advisory panel vote against use in type 1 diabetes, while safety concerns for type 2 diabetes were not raised. The safety profile is generally consistent with other basal insulins, featuring adverse events such as hypoglycemia, injection site reactions, lipodystrophy, itching, rash, swelling, and weight gain. Awicli enables a once-weekly regimen that could reduce injections from 7 per week to 1, administered on the same day each week using the Flex Touch pen. Awicli is the first once-weekly basal insulin approved for adults with type 2 diabetes and is now broadly available in the United States. Dosing guidance emphasizes correct device use, site rotation, and safety precautions to avoid dosing errors. International approvals include the United States, European Union, and 13 other countries, underscoring accessibility and patient support initiatives. FDA approval in March 2026 was based on the Onwards program, comprising 5 Phase 3 trials that showed non-inferiority in HbA1c reduction compared with daily basal insulin across diverse adults with type 2 diabetes. Across 4 Onwards trials involving 2,600 adults, once-weekly icodec demonstrated non-inferiority or superiority in glucose lowering when used with various regimens, including mealtime insulin, a GLP-1 receptor agonist, or oral agents. Awicli is available nationwide at over 70,000 retail pharmacies, with affordability programs through novacare.com that can reduce out-of-pocket costs to as little as $35 per month for eligible patients. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.